Alright, I'm going to try to explain DNA replication to you using the textbook chapter, The Molecular Basis of Inheritance. It is on Moodle, it's in the textbook Campbell Biology, and you can see up here the title of the textbook chapter so you can find it easily. Before I go into replication, I'll start out with the basics of DNA structure. So DNA is made up of two strands of nucleotides, and this is what forms the double helix. A nucleotide is a phosphate group bonded to a sugar, which is bonded to a base, a nitrogenous base. The rungs of the double helix are what we call base pairs, and each of those base pairs contains two bases hydrogen bonded to each other. Now this is what we call complementary base pairing, because one base will only hydrogen bond with one other kind of base. So adenine only hydrogen bonds with thymine, and guanine only hydrogen bonds with cytosine. And that has implications for DNA replication. What also has implications for DNA replication is the fact that the DNA molecule, or the strands of the DNA molecule, are what we call anti-parallel. So that means on one side of the DNA molecule, the nucleotides run in the 5' to 3' direction, and on the other side of the DNA molecule, the nucleotides run in the 3' to 5' direction. So they're oriented in opposite directions. Just so you know, the terms 5' and 3' refer to the different carbons on the sugars. You don't really need to know any more than that. I'll explain in a bit why that anti-parallel nature of DNA, the fact that the strands run in opposite directions, is significant. So how does DNA replication actually work? Well, here are the basics. We start with a parental DNA molecule. The hydrogen bonds between the base pairs are broken, and then in this space here now we can have two daughter strands that come in, and those daughter strands are built by using complementary base pairing. So an enzyme comes along and it looks at one of the parental strands and it goes, okay, well the sequence here is A, C, T, A, G, and I know that A only bonds with T, so I'll get a T, and I know that C only bonds with G, so I'll get a G, and so on. And that's how the daughter DNA strands are built, and the same thing happens on the other side. And so eventually this ends up with two daughter DNA molecules, and each of those daughter DNA molecules contains one parental strand of DNA and one daughter strand of DNA, one new strand of DNA. And we call that the semi-conservative model of replication. And we call it semi-conservative because half of the parental DNA molecule is preserved in each of the daughter DNA molecules. Now, where does this new DNA that makes up the daughter DNA molecules come from? Well, it comes from the food that you eat, because pretty much all the food you eat contains DNA, and the nucleotides in that DNA are pulled apart and then used to build new DNA inside your cells. So here we can see the semi-conservative model. We have the two parental strands of the parent DNA molecule being preserved and lined up with two new daughter strands, so that each new daughter DNA molecule has one parental strand and one daughter strand, and that's why we call it the semi-conservative model of replication. Now this model of DNA replication has been shown to be correct as opposed to the conservative model and the dispersive model, which have been shown to be incorrect, and it was an experiment by Michelson and Stahl, a pretty cool experiment actually, which showed that the semi-conservative model was the correct model of DNA replication, and you can read more about that in this textbook chapter. So, how does DNA replication work? What happens is there are many sites along a eukaryotic chromosome, and we're eukaryotes with linear chromosomes. There are many sites along the chromosome called origins of replication, and that's short base sequences along that eukaryotic chromosome. An enzyme recognizes an origin of replication, and at that point it unzips the DNA molecule forming a replication bubble. And within that replication bubble, we can get the two daughter strands matching up to the two parent strands to eventually form two daughter DNA molecules. So what happens inside a replication bubble is an enzyme comes along and starts banging nucleotides together to form the daughter strand, and replication proceeds in the direction of something called a replication fork, which is the point at which the DNA molecule is beginning to be unzipped. So basically these bubbles, which start quite small, eventually widen as the replication forks on either side of each bubble move in opposite directions. And as those bubbles are widening and the daughter DNA strands are being made, eventually those bubbles meet up and we end up with two daughter molecules, each of which have one parent strand from the original DNA molecule and one daughter strand made up of new DNA, which has been put together by enzymes. So what enzymes are involved in this process? Well, we start out with an enzyme called helicase, which comes along and unwinds and unzips the parental DNA molecule at the replication fork. That creates the replication bubble, and to stop this bubble snapping back together, because obviously these two strands of the parental DNA molecule are attracted to each other because the bases are attracted to each other, to stop those two strands snapping back together, we have something called single-strand binding proteins, which come along and stop those strands from coming back together. Now, because pulling apart those two DNA strands also creates a bit of pressure, a bit of tension ahead of the replication fork, we need another enzyme to come in called topoisomerase, and it basically relieves that tension ahead of the replication fork, and it does that by breaking, swiveling and rejoining DNA to stop there being too much pressure here. Now, once the replication bubble has been formed at a replication fork, DNA replication proceeds this way as the bubble progressively widens. So the way that works is something called primase, another enzyme, first has to lay down something called an RNA primer, which is a sequence of RNA nucleotides, a very short sequence. And the reason it does that, and it matches those nucleotides up to the parental strand, by the way, but the reason it does that is because DNA polymerase 3, which is the enzyme that actually makes the daughter DNA strand, can only attach nucleotides to the end of a pre-existing strand of nucleotides. That means it can't come in and start the process without having a strand of nucleotides to attach its nucleotides to. So that's what the primase does. It makes an RNA primer so that the DNA polymerase 3 actually has something to attach nucleotides to. But once it comes in and the RNA primer is there, it can just do its thing. It works its way along the DNA, attaching nucleotides one after the other. But DNA polymerase 3 is fairly limited. It can only attach nucleotides in one direction along the DNA molecule. It can only move in the 5' to 3' direction. And because DNA is anti-parallel, meaning that one strand goes 5' to 3' and the other strand goes 3' to 5', that creates some issues. So let's just have a look at this diagram here. Let's say that this DNA molecule, this parental strand, runs from 3' to 5'. That means that this daughter strand must be 5' to 3' because DNA is anti-parallel. Because DNA polymerase 3 works in the 5' to 3' direction, that's fine. It can just gradually add nucleotides one after the other in the direction of the replication fork as that bubble widens, and it doesn't have any problems there. But this strand down here, that creates some issues. Because if this parental strand is 3' to 5', then this parental strand must be 5' to 3'. And that means that this daughter strand must be 3' to 5'. So the DNA polymerase 3 here can't work this way to create that daughter strand. It has to work in the opposite direction, this way. But obviously that creates some issues, because if this strand is being made continuously this way, and we call this the leading strand, then this strand can't be made continuously this way in the opposite direction, because this strand has to keep up with this strand here. So hopefully you can see why that could potentially create some problems. So the upshot of that is this. A strand called the leading strand, one of the daughter strands, can be made continuously. So the primer is put down by RNA primase, and then DNA polymerase 3 just comes along and it just bangs on the nucleotides one after the other in the direction of the replication fork as that bubble widens. The other strand, on the other hand, has to be synthesized in the opposite direction. But because this other strand has to keep up with the leading strand, this other strand can only be synthesized in a series of fragments. So the way it works is like this. We've got the leading strand being made continuously, and then the lagging strand is made 1, 2, 3, 4, 5, 6, etc. And it's made in those fragments so that the DNA polymerase on this strand can keep up with the DNA polymerase on the leading strand. So these fragments are made one after the other. And those fragments are called Okazaki fragments. Now, Okazaki fragments each have to start with an RNA primer, because DNA polymerase works that way. It needs an RNA primer to start creating the daughter DNA molecule. So we have a series of fragments, each starts with an RNA primer, and then we have a sequence of DNA that's put there by DNA polymerase. So at the end of the entire process, once all the DNA has been made, we need to get rid of those RNA primers, because RNA is not DNA, and obviously we're trying to make DNA molecules. So something called DNA polymerase 1, which is a completely different kind of DNA polymerase, takes out the RNA primers and replaces them with DNA. And then we have another enzyme called DNA ligase, which comes along and links all those little fragments of DNA together on the lagging strand to make a complete continuous DNA strand. So to summarize that entire process, we have helicase, it unwinds the double helix. Then we have single strand binding proteins, which keep the two parental strands apart from each other. And then we have RNA primase, which on the leading strand simply lays down an RNA primer, and then DNA polymerase 3 comes along and synthesizes that leading strand in the direction of the replication fork. And on the lagging strand, we have fragments called Okazaki fragments, which are made one after the other in the opposite direction to the leading strand. And the reason the lagging strand is made in fragments is because the DNA polymerase there is trying to keep up with the leading strand, which is moving further and further along the replication fork, or sorry, towards the replication fork as that bubble widens. So in the end, the lagging strand is a series of fragments. What we need to do is get rid of the RNA primers at the beginning of each of those fragments. So it's DNA polymerase 1 that does that, replaces those RNA primers with DNA. And then it's DNA ligase that comes along and links all the fragments of DNA together to make a continuous strand. And as a result of that entire process, we end up with two daughter DNA molecules. Each of them has one parental strand from the original DNA molecule and one daughter strand, which has been made in this process.